This lecture addresses one of the most important questions to arise from the pandemic. What is the origin of SARS-CoV-2? I'm going to present genetic evidence that seems to suggest that the lab-leak hypothesis and the idea that SARS-CoV-2 did not naturally evolve are the most parsimonious explanations for where the virus came from. The lab leak or non-natural evolution concept has been highly controversial, and it's led to some alarming examples of censorship, which is my motivation for making this lecture. I really want to push back on censorship and encourage open discussion. So in order to get an understanding of how the virus evolves, we got to look at its replication mechanism. And so what you can see here is the template, which in this case is the virus's genome, and this replication complex of proteins, which is making copies of the genome so that the genome can ultimately be translated into proteins. And so you see these non-structural proteins, 7 and 8, that glom onto the RNA polymerase, the RNA-dependent RNA polymerase, NSP12, and facilitate essentially copying this template into a product. In addition, there's a helicase which helps unwind so that the polymerase can read through and create the copy strand. The other thing that's present is this NSP14. NSP14 is a proofreading enzyme that has 3', 5' exonuclease activity, so it actually minimizes the mistakes that SARS-CoV-2 makes. But still, SARS-CoV-2 makes a lot of mistakes. NSP10 adds this cap so that the nascent strand that you see here, when it's finally done, can be translated into proteins, makes it basically look like human messenger RNA, and therefore it can be translated into proteins, in this case virus proteins. This slide just gives you a sense for the 3', 5' exonuclease concept, and again here they're using DNA, but it still helps to illustrate that concept. Again, NSP14 is that protein that I talked about that SARS-CoV-2 has for error correction. So imagine if this is the template, and these are the bases that make up the template, and here's the complementary strand that the polymerase is laying down. In this hypothetical example, the polymerase has made a mistake. It's supposed to put a G complementary to this C, and in this particular case, it throws in an A. So rather than just reading through and leaving these mistakes behind, this 3', 5' exonuclease activity allows the polymerase to move backwards and cut that out and then replace the base with the correct one. And so by doing this, in this particular case, the virus minimizes the likelihood of building up detrimental mutations that would result in that particular lineage going extinct. But again, still, despite having NSP14, the virus still makes mistakes, as we can see from this slide. These are some examples of changes in the genomes of some of the variants that have arisen. Again, point mutations giving rise to variants, and not just point mutations, but deletions as well. Here is a schematic representation of the amino acids that make up the spike protein of SARS-CoV-2, and then some important regions highlighted, such as the receptor binding domain. The amino acids for that fall in this region here. And another one is this Furyn cleavage site, this tiny little sliver here that allows the virus to penetrate human cells. The receptor binding domain allows the spike protein of the virus to dock onto those human cells. And so again, here's the alpha variant, for example, and it has a specific deletion here, another deletion here of this tyrosine. And then it has some point mutations where the virus was supposed to put an asparagine amino acid here at position 501, but instead put a tyrosine there. And so there are these mutations that occur in this receptor binding domain, and some of them are advantageous and give these viruses the ability to bind better and therefore transmit better. The beta variant, for example, the so-called South African variant, also has an array of changes that were made, and you can see those. Now, one thing I should point out is that very few of these, and all the variants that have been produced, have any changes or significant changes to the Furyn cleavage site, and that's going to be an important concept that I'm going to come back to. This slide depicts some transmission patterns that have been established for two of SARS-CoV-2's close relatives, MERS-CoV and SARS-CoV-1, and then presents a hypothetical transmission pathway to humans for SARS-CoV-2. So MERS, for example, most likely originated in a bat and used a camel intermediate host, which then transmitted to humans. And in this part of the world, in the Middle East, lots of individuals come in regular contact with camels. They ride them and they eat them. And so there's lots of opportunity for camel respiratory secretions to infect humans. This particular virus is very lethal and often burns out pretty quickly in the human population. Similarly, SARS-CoV-1 presumably originated with a bat and then followed the pathway through an intermediate host of a civet cat and then ultimately to humans. Now, civet cats are eaten, but they're also used in a weird civet cat coffee. So there's lots of opportunity for individuals in certain parts of the world to come in contact with civet cats. With SARS-CoV-2, it's a little bit more complicated. Now, the theoretical originator came from a bat, and in this pathway shows a pangolin, and then some either intermediate host or a direct transmission from the pangolin, and that doesn't seem to be logical, the direct transmission. The point is that there's enough difference between SARS-CoV-2 and the viruses taken from these animals to suggest that something else came here. So again, an unknown animal, and I have to point out that thousands and thousands of animals have had their coronaviruses sequenced by researchers, and nothing like SARS-CoV-2 has been discovered. A more parsimonious explanation is some sort of a lab leak in which there was artificial evolutionary pressure placed on some progenitor virus, which resulted in the formation of SARS-CoV-2. And so if you get a chance, you can watch this particular YouTube video, and I'll address this paper that's discussed here by the lab of Dr. Nikolai Petrovsky, in which Petrovsky just makes a compelling argument for a lab leak. And so here's the paper, and the paper basically is an in silico comparison. So what that means is using computer modeling of SARS-CoV-2 spike protein ACE2 binding affinities across a variety of species and its implications for virus origin. And so again, what that means is they mapped the spike proteins of some of the early isolates of SARS-CoV-2 and then aligned them with receptor proteins from a variety of different animals, and their conclusions were as follows. These findings show that the earliest known SARS-CoV-2 isolates were surprisingly well adapted to bind strongly to human ACE2. Spike protein exhibited the highest binding to human ACE2 of all species tested. These data suggest that SARS-CoV-2 spike receptor binding domain evolved by selection on human-like ACE2. And so what they're saying here is that it's very unusual that a virus taken from a human so recently would be best adapted to the human. In fact, if the virus came directly from the pangolin, the virus should have the most affinity to pangolin receptors. Or if it was very recently from a bat, it should have receptors more apt to bind to bats. But in this particular case, SARS-CoV-2 was human adapted from the very beginning, which is not a typical thing in evolution. If you want to read a little bit more about that, here's this article I was interviewed for along with Nikolai Petrovsky. Another compelling piece of circumstantial evidence that leans towards the likelihood of a lab leak is that SARS-CoV-1 escaped from labs on at least two occasions, resulting in the deaths of individuals. And the explanation for that was breaches in safety regulations, individuals working on these viruses, but doing so in BSL-2, for example, as opposed to BSL-4 or higher level biosafety level procedures. Another extremely strong piece of circumstantial evidence came from this 2017 paper. A SARS-like WIV1, so WIV stands for Wuhan Institute of Virology, so it's interesting that the virus originated in Wuhan. The first cases were in Wuhan, and coincidentally or not, there is a Wuhan Institute of Virology that, according to this paper, was actually working on these so-called pre-pandemic strains. This manuscript describes efforts to extend surveillance beyond sequence analysis. They were constructing chimeric and full-length zoonotic animal-infecting coronaviruses to evaluate their emergence potential. That's essentially gain-of-function research. They're creating strains of coronavirus with enhanced ability to infect, in this particular case, humanized cells and humanized animals. That is, mice, for example, that express human receptors. Together, the data highlight the utility of a platform to identify and prioritize pre-pandemic strains harbored in animal reservoirs and document the threat posed by WIV1-CoV for emergence in human populations. So to reiterate, they are creating these virus strains and then studying their ability to infect human type cell culture and humanized animals in the laboratory to understand their potential for emergence in the population at large. Another interesting aspect of the paper is they recognize that it's not just the binding, the S1 or the S1 region of the spike protein, which binds to the human receptors. They're also looking at differences in other regions, such as regions that can result in enhanced spike cleavage, which in turn would give expanded tropism for different cell types, including humans. And so that becomes an important concept later on when I talk about the Furyn cleavage site. So in order to get a better understanding of all this, you need to look at a schematic diagram of the SARS-CoV-2 spike protein. So it does have a receptor binding domain that binds to human ACE2 on the surface of cells. ACE2 is part of the renin-angiotensin system that's essential for regulating blood pressure. And so it's not evolved in humans to bind to SARS-CoV-2. So it does have a natural function, but SARS-CoV-2 has co-opted it using this RBD receptor binding domain region in order to bind to it. But what's important here is another element that's important here is the fusion peptide. So once the receptor binds, the receptor has to be clipped in order for this fusion peptide to be exposed to the cell surface and essentially pry the virus into the cell. It's essentially like trying to catch an elevator, and as the doors are closing, you put your hands in and then you pry the doors open. And that's what this fusion peptide's job is, to pry the doors open so that the virus can get in. But it requires a cleavage here in order to function. And so if we look at this particular cleavage site, the amino acid sequences of SARS-CoV-2 next to SARS-CoV-1, there's some similarities, but there's a vast difference right here in the so-called Furyn cleavage site. SARS-CoV-2 has these amino acids here that a lot of viruses, that none of the viruses that are closely related to it have in this particular location. This P, R, R, A, and then the R is in common, but these other arginines, which become really important in Furyn cleavage, are not present in these other closely related viruses. So that increases the efficiency with which this site is clipped so that the fusion peptide can be exposed and allow the virus to get in. The more R's that are here, the more likely the furin is to cleave this, and the more likely the virus is to get into cells. Another important concept I wanted to point out is how these R's are encoded in the genome. These key R's right here, how they're encoded in the genome. And in order to understand what I mean by that, you have to have a basic understanding of the central dogma in molecular biology. Now, in animals and microorganisms that have DNA as a genome, DNA basically replicates and makes new copies of DNA, but we're talking about a virus which has an RNA genome. So let's start here. SARS-CoV-2 has an RNA genome that can be translated into proteins, and then these proteins are the spike and all the rest of the things that make up the virus. So within this RNA are short little coding sequences, these little 3-base coding sequences called codons. And I want to specifically point out the codons that encode the arginines, the R in that diagram here. This is an abbreviation for arginine. There are actually 6 codons that encode arginine. As you can see here, these 4 and then these 2. And the individual organisms will prefer certain codons to make arginine. And so a simple look at the codon usage in SARS-CoV-2 points to something very interesting, and that is that this codon, CGG, encoding arginine in SARS-CoV-2 is extremely rare. And that becomes an important point in understanding whether or not this might be a lab leak. And I'll get to that in a second. So if we look at ORF1A, for example, this is a big region of the SARS-CoV-2 genome. It's 13,217 base pairs long. So over 13,000 of these individual chemicals long. But of that 13,000, only 3 codons, so 9 bases total, are CGGs. So 3 out of 13,217. In ORF1B, which is smaller but still a really big section of the genome, it only uses CGG 4 times to encode arginine. In the nucleocapsid, it only uses CGG twice. And in the membrane glycoprotein, it doesn't use CGG at all. The virus much prefers to use AGA, CGT, and other codons over CGG. If we look at the spike, however, those 2 contiguous R's that you see here are both encoded by CGGs. So now we have this astronomically rare codon right next to each other in duplicate. This slide is intended to give you a close-up of the spike protein RNA sequence, specifically encoding the Furyn cleavage site. And so what you see here is the 5' to 3' organization of the genome. And here, this big green box in the middle is the stretch of RNA that encodes the spike protein. So this region here would be translated into a protein that would fold up and make this spike protein. And then somewhere in the middle here is the Furyn cleavage site right here. This is where the host-derived proteases would cut the spike protein, exposing the fusion peptide so that the virus can insert itself into the host cell. As I've talked about, and we'll talk more about, the Furyn cleavage site pictured here, the PRRAR amino acid sequence that's encoded by this particular stretch of RNAs, greatly enhances the ability of the virus to infect. and transmit. And so these key CGG codons that are pictured here, the ones that I've said that are extremely rare, that happen to be contiguous in this one location, in this very one key location in the genome, is surprisingly suspicious. Another interesting aspect of this, though, is that the Delta variant, which is the new dominant variant of concern that's caused a lot of problems in the last few months, actually has a mutation here. So that the codon was CCU encoding a proline, now is CGU. So this C changed to a G in the Delta variant, and that encodes arginine. So now the Delta variant Furyn protease is RRRAR. Again, showing you the importance of these multiple arginines in this particular cleavage site. When I presented these data to a structural chemist who is a leading researcher on vaccine development in the world, he had said specifically, human gene jockeys commonly use CGG to encode arginine. So when they want to insert arginine into a virus genome, they'll use the codon CGG to do that. It's a preferred codon. And again, the fact that there are two of these very rare CGGs right next to each other in this highly important Furyn cleavage site is highly suspicious. So this table, taken from this publication, is really important because it illustrates a couple important trends. This is Furyn cleavage probability of spike sequence homology. And so again, we've got the description, so the particular virus, and then the actual Furyn cleavage site sequence. And these are amino acid sequences. And then the Furyn score. If the score is above 0.5, then it's a strong Furyn cleavage site. Identity is how close the virus is related in terms of its spike to SARS CoV2. So these top viruses here, obviously SARS CoV2 is a 100% match to itself. And then this bat virus, RATG13, is very closely identical to SARS CoV2. This other bat virus is also fairly highly identical. And then SARS-CoV-1 is another example. So what's interesting is that all these viruses that have the closest identity, or rather highest identity to SARS-CoV-2, also have the lowest Furyn scores. And so you can see here the PRRAR, the presumed Furyn cleavage site, is a pretty high score. We don't find these high scores until we get into these viruses that are very dissimilar from SARS-CoV-2. And I pointed out this one in particular. Here's a human common cold coronavirus, HKU1, that's not very similar to SARS-CoV-2 at all, but happens to also have one of these really robust Furyn cleavage sites. So one explanation for this is that individuals trying to create a endemic, highly transmissible virus took the liberty to add some of these arginines to the SARS-CoV-2 Furyn site here, spike protein, to make a Furyn cleavage site. That's one explanation, and there are others, but that's a pretty parsimonious one, if you ask me. And so if we look at the flanking amino acids around the Furyn cleavage site, you can see that there's a fair amount of alignment here between some of these really closely related coronaviruses to SARS-CoV-2. I threw the pangolin in here, but here's SARS-1 and RATG13 that appear on this table. But the pangolin virus is new to this. And again, just so you can see that there's a lot of similarity, at least in terms of the flanking regions immediately around the Furyn site. But again, here's the Furyn site that I've underlined, and there's nothing like it in the other viruses. So the question again is, did they, recognizing the importance of arginines, artificially place these here, or did evolution somehow place it there on accident? Again, using CGG twice in a row, where that doesn't occur anywhere else in the nearly 30,000 base pair genome. If we look at that, if we look at this from a wider taxonomic perspective, comparing SARS-CoV-2's Furyn cleavage site with a variety of other viruses that are taxonomically different, each of these yellow branches, each of these colored branches represents unique taxons. And so some of the farthest taxonomically from SARS-CoV-2, this group here has pretty robust Furyn cleavage sites. Here's a human common cold virus, OC43. Here's HKU1, another common cold virus. And again, they're endemic viruses that we cannot get rid of. And they have multiple arginines in these particular locations. Notice again that this particular clade, this particular group of viruses that are closely related, of them, only SARS-CoV-2 has what we call the polybasic Furyn cleavage site, meaning polybasic, meaning that it has multiple arginines. These guys don't have anything like that. They have the single arginine, which may give it some ability to be cleaved with some level of efficiency, but nothing as nearly as efficient as having multiple arginines there. And again, it's about efficiency. It's not about yes or no. This does happen or it doesn't happen. The cleavage does happen or it doesn't happen. It's about efficiency. And you don't need much improvement in efficiency if you're a highly replicating virus to really gain a significant advantage. This paper in perhaps one of the most prestigious microbiology journals on the planet illustrates my point about the importance of Furyn cleavage. And so the title, "The Furyn cleavage site in the SARS-CoV-2 spike protein is required for transmission in ferrets," only tells part of the story. One of their conclusions, we show that the polybasic insertion, so again, multiple arginines, endows SARS-CoV-2 with a selective advantage in lung cells and primary human airway epithelial cells. SARS-CoV-2 viruses that lack this Furyn cleavage site shed lower titers, meaning that they don't produce as many progeny from infected ferrets. And ferrets are a great model for this particular virus. Obviously, they're not going to use people and do it that way. So in infected ferrets, they shed fewer viruses when they don't have a Furyn cleavage site and was not transmitted to co-housed sentinel animals. So again, you put animals together, you infect some, you co-house them with sentinel animals, and then you look for transmission. And that was not possible in the absence of Furyn cleavage sites. This is another important point that was made in the paper. Analysis of 100,000 SARS-CoV-2 sequences derived from patients with 24 human postmortem tissues showed low frequency of naturally occurring mutants that harbor deletions at the polybasic site. And what that tells us is that if the virus starts making tons of mutants in that short little region, it basically goes extinct. So it's that important. And therefore, again, it's a super important region of the genome and of the spike protein. And therefore, the virus would not be motivated, evolutionarily speaking, to make changes to it. Taken together, our findings reveal that the Furyn cleavage site is an important determinant of SARS-CoV-2 transmission. So this diagram gives you an overview of how SARS-CoV-2 infects and what needs to happen for it to efficiently transmit. And the virus, as you can see here, touches down and initially looks like it binds to high levels of sialic acid, these sort of sugar groups that are on the surface of most cells of our body. And by grabbing onto these loosely, it can then form more tight binding to these protein receptors. Ultimately, this SARS-CoV-2 spike protein is clipped And then that facilitates the fusion peptide interacting with the membrane so that the virus can fuse with the cell and then inject its genetic information into the cell. The genetic information is then copied and translated into proteins, and then viruses are made that are exocytosed into the world to infect more people or to infect neighboring cells. But there's another component to this that SARS-CoV-2 relies on, syncytia formation. So neighboring cells can fuse in the presence of this virus. And the reason for that is the virus's spike proteins can cause it to fuse, can basically zipper it to the neighboring cell. And that creates a bridge so that the virus's genetic information can directly pass into the cytoplasm without having to go through this process of forming a full virus and going into the world. This enhances efficiency of spread from cell to cell in your body astronomically. And this particular paper here highlights the importance of the Furyn cleavage site in syncytia formation, in the formation of these cell-cell bridges. And so here are the data, and this graph is just showing you syncytia formation on the y-axis, percentage. So they're looking under the microscope and they've exposed cells to these different strains of virus and are then looking under the microscope and counting the percentage of syncytia formation formed in various cells. This over here just shows you the genetics of the different strains. So I'll focus specifically on SARS-CoV-2. So here's wild type SARS-CoV-2 here with its 3 arginines in the polybasic Furyn cleavage site. When they created a mutation in SARS-CoV-2, so this is a mono-CS, meaning that they took SARS-CoV-2 and they removed 2 of the arginines and filled them with other amino acids. Notice how there's a significant drop in syncytia formation. Conversely, when they added arginines in this H5CS strain, and so now they've got 5 arginines added, 1, 2, 3, 4, 5 in the polybasic cleavage site. It goes up astronomically again. So the syncytia formation is proportional to the amount of arginines present. Here's the delta CS mutant. So the delta CS mutant was literally, they just cut that completely out of SARS-CoV-2 and you lose syncytia formation. What's also important though is the flanking region. So basically in delta flank here, they took just the flanking amino acids away and they left the PRRAR polybasic Furyn cleavage site. So it shows you that it's not just this that's important, but it's also these flanking amino acids. So again, taken together, our findings reveal that the Furyn cleavage site is an important determinant of SARS-CoV-2 transmission. This slide is intended to show you the importance of the Furyn cleavage site in SARS-CoV-2. Over here on the left, you see SARS-CoV-1 binding to the ACE2 receptor and using human-derived trypsin and a serine protease in order to mature its spike protein, enter the cell, be trafficked in these intracellular vesicles, and at various points along the way using human cathepsin to further mature the spike proteins and facilitate release of the genetic information into the endomembrane system and then eventually form full viruses and kick out into the world. Something important to note though is that SARS-CoV-2 was essentially a self-limiting pandemic. The virus essentially disappeared after a period of time. And obviously there were mitigation strategies put in place, but there were mitigation strategies put in place with COVID-2 and it's not going anywhere. And so a strong hypothesis to explain this is the lack of Furyn cleavage, the lack of that extra efficiency that we see here with SARS-CoV-2. SARS-CoV-2 can use trypsin and the serine protease, but also has the ability to be matured by the Furyn protease that humans make. So the virus binds to ACE2, and these different proteases can work together to mature the spike protein to allow the virus to release its RNA into the cytoplasm. At various stages here in the endoplasmic reticulum and here in the Golgi apparatus and these vesicles that bud off of the Golgi, the virus is constantly being matured by Furyn cleavage and essentially being released into the world as a much more infectious virus. These data reveal the importance of Furyn cleavage, specifically the importance of having multiple arginines in your Furyn cleavage site. So here's SARS-CoV-1, and this is a size scale over here, and this is the uncut spike protein. The uncut spike protein should make a big, thick band at this, roughly 200 or so kilodalton region of this gel. And so when you purify the spike protein and run it on this gel, if the spike protein isn't cleaved or is minimally cleaved, you'll see a big, heavy band here showing where all those proteins end up in the gel. And again, because SARS-CoV-1 doesn't have a strong spike protein, when these spike proteins are exposed to Furyn protease, very little cleavage takes place, if at all. There's a faint band there. With wild-type SARS-CoV-2, most of the spike is cleaved. That's why you see this bigger band here showing much higher proportion of spike proteins being cleaved, and then a smaller band here. Again, it's not 100% cleavage, but clearly it favors cleavage and the formation of the smaller spike. When you create a Furyn cleavage site with only a single arginine in it, most of the spike remains unprocessed, and very little cleavage happens. And again, cleavage is proportional to infectivity. The ability of that spike protein to be cleaved is essential for the virus to get into the cell and do undergo this replication cycle. In this particular example, this is a strain of SARS-CoV-2 that has H5CS. And so again, if I back up and show you the H5CS, this is a version of the virus in which 5 arginines were added. And you can see that almost the entire spike protein population is cleaved into the fully infectious form. And then if you remove that entire cleavage site, nothing is cleaved. Similarly, if you remove the flanking amino acids, most of the spike remains uncleaved and very little of it gets cleaved. And so these data support the hypothesis that, number one, that the more arginines present in the Furyn cleavage site, the more efficient the cleavage, and therefore the more efficient the infection cycle of the virus. When you take this genetic evidence together with some of this other circumstantial evidence and very suspicious things that have happened in Wuhan during this time frame in the past year, it again points to a certain conclusion. For example, deleted coronavirus genome sequences trigger scientific intrigue. So we know that researchers from the Wuhan Institute of Virology deleted a bunch of coronavirus genomes from the database. Now, this happens. Sometimes we make mistakes and we want to delete things and then fix them and re-upload. But I don't believe that the sequences have ever been re-uploaded. So again, taking away key evidence from the crime scene under normal circumstances is a really serious offense. Here's another article. Wuhan lab has hidden more on COVID-19 than it has revealed. So in this particular case, they took this particular virus, Ra-BT-CoV-4991, which was collected in Yunnan province, which is thousands of miles from Wuhan, was from the same copper mine shaft in which 6 miners had developed ARDS. 3 of the miners died. And so this is significant because what was discovered later on was that this virus, which is alarmingly similar to SARS-CoV-2, killed some of these individuals back in 2013. They changed the name of this virus to Ra-TG13, in effect trying to destroy the paper trail between this lethal SARS-CoV-2-like virus by renaming it something else so that this virus no longer had the stigma of having killed somebody attached to it, and the fact that it was very similar to SARS-CoV-2. Again, very suspicious. So the question remains, what is the origin of SARS-CoV-2? Was this an accidental lab leak of a CoV-2 strain optimized by passage in humanized mice or cells? The data from Nikolai Petrovsky suggests that the virus at the very beginning was optimized for binding to human ACE2. So this bullet point is strongly favored by those data. The second bullet point was an accidental lab leak of a deliberately genetically modified SARS-CoV-2 strain. And when we look closely at the Furyn cleavage site, this suggests that perhaps it was. The CGG codon usage and the lack of Furyn in closely related viruses to SARS-CoV-2 suggest that it was deliberately genetically modified. Or was this a natural transmission from an animal in the wet market or whatever? And did those CGGs just happen to come together through natural combinations and natural evolution, resulting in the formation of that Furyn cleavage site that gave this virus a tremendous advantage and the ability to essentially become endemic and defy all our mitigation strategies? Or is there another scenario? Another important question is, what are the ramifications of each scenario? Obviously, there are huge political ramifications for the Chinese government if the virus accidentally leaked from the Wuhan Institute of Virology. But there's also significant ramifications for politicians all over the world for the contentious mandates that they implemented, polarizing populations. And of course, there's huge financial ramifications, particularly for vaccine makers that are making billions of dollars off of the pandemic, especially if science was the cause of the pandemic. And another big ramification is just an overall loss of trust in science. There's a bit of that. There's quite a bit of that, actually, an erosion in trusted science, but this would absolutely decimate trust in science. What additional data do we need? Well, the main thing that we would need is some kind of missing link discovery. We know that SARS-CoV-1 has jumped from civet cats by way of bats into the human population. And we also know that MERS occasionally jumps from camels into the human population. And so something similar, a similar scenario with SARS-CoV-2 would obviously go a long way to supporting the natural transmission hypothesis. It would also be great to get some of the lab data, but I don't know if the Chinese government and specifically the researchers at the Wuhan Institute of Virology will be very forthcoming with that kind of data, and especially the stuff that they deleted. And so that may be a trickier scenario in which to support the hypothesis that this leaked from the lab. We definitely need more individuals, more credible individuals to come together and really look at this problem, because it is the most important question in the pandemic. What is the origin of SARS-CoV-2?